Now, in science and engineering, we have things called the national academies. So if you're in the National Academy of Science, it's like you are like a Hall of Famer in science. If you're a National Academy member in medicine, you're a Hall of Famer in medicine. If you're a National Academy member, you're a Hall of Famer in engineering, you're a Hall of Famer in engineering. And she is in 3 halls of fame. All at once. Basically on one of 2 hands, the number of people in this country who are in all 3 national academies. And that is really quite a feat. So we're really, as a colleague, we are so proud to always have Paula on our faculty here at MIT, and she's been just a leader in all things around the Institute. I can't even just kind of go into the number of things that she's done that have improved the Institute over her years here. And she's also, as you can see, just like an amazing scientist. And we're going to hear about her journey as a science tonight.
Thank you so much, Tim. Thanks so much. That was a great introduction. I am really excited to talk to all of you today. So I will dive right in before the dinner sets in and everyone gets sleepy. I'm going to talk about nanoparticle systems that we design in our lab using electrostatics that allow these nanoparticles to have properties that are really interesting for drug delivery to tumors. And to start out, I should say that these ideas came from work we've been doing earlier. The idea of absorbing electrostatic layers, layer by layer, is a simple one. You start with a surface that has, let's say, a negative charge. You immerse that into a dilute water solution that has a positively charged polymer, and that polymer will absorb onto that negatively charged substrate until ultimately you reverse the surface charge. Now you have something that is same charged, like charged, and therefore nothing else deposits. So you have a single layer that absorbs. You can rinse that film and remove anything that's not truly electrostatically absorbed. And now you can absorb the oppositely charged layer again until the surface charge is reversed. So this process of building positive and negative layers on top of each other in this thermodynamically controlled way has been termed layer by layer. And that's been around now for a few decades. We decided to look at how we might be able to use that approach to modify nanoparticles that are used in drug delivery. And this idea is based on the fact that you can use a nanoparticle, something that is small enough to be delivered in the bloodstream and can be designed so that it ultimately finds its way to the cancer. Now, the work that we did started with looking at liposomes and also with polymeric, degradable polymeric nanoparticles, with the idea that these are vessels that can be stuffed with chemotherapy drugs. And there are already some FDA-approved examples of that. Doxorubicin is one of them. It comes in the form of Doxil, which is a well-known cancer drug. So we know we can stuff a cancer drug inside, for example, the aqueous core of a nanoparticle like this one. We also know that there are a lot of cancers that don't really succumb to the presence of that chemotherapy drug. We really want to be able to enable that drug to be effective. And it turns out that a lot of cancers have genetic mutations that allow them to survive even in the presence of chemotherapy. So in our earliest work, we began looking at how we might be able to take a liposome and stuff a chemotherapy drug in the core of it. If we design that liposome so that it is negatively charged by introducing some negatively charged lipids into the liposomal layer, then we can absorb a positively charged polymer on top of it. And we can follow that positively charged monolayer with siRNA or DNA or some other nucleic acid. All nucleic acids are negatively charged. And siRNA, silencing RNA, can actually silence a gene that is enabling the tumor to survive in the presence of chemo drug. So the idea is let's incorporate siRNA that will silence that tumor's defense mechanism against the drug. And if we layer it this way, the first thing that will come out is the siRNA. silencing the defense mechanism. And the second thing that will come out is the chemotherapy drug, enabling a more effective killing of the cancer cell. siRNA is a delicate molecule in the sense that it degrades rapidly in the presence of the RNases that are in our bloodstream. So we need to protect it, and so we put down another positively charged layer, and now we have the siRNA in a sandwich of plus, minus, plus. And that gives us sufficient protection against the enzymes that might break it down. Because these are really highly interwoven, we draw them as separate lines because, you know, it takes so much work to create what this really would look like, but these are really, these polymeric coils intertwined with each other. So really they would be blurred a little bit. Finally, if we were just to introduce a positively charged nanoparticle into our bloodstream, it would rapidly absorb all of the negatively charged proteins. There are a large number of especially negatively charged proteins in our bloodstream, and be recognized as foreign matter that needs to be eliminated. And our immune system would flush it out. So it would have a very short time in the blood before it got removed. And that means that we need to protect that nanoparticle from being eliminated as rapidly so it has time to get to the tumor. So instead of sending in a positively charged nanoparticle, we make it negatively charged. And the advantage to this is that a large number of mammalian cells have glycans stuck to their membrane that make them negatively charged too. So it gives us a little bit of self-repulsion, that the nanoparticle isn't going to interact without some other sort of encouragement with other cells. At least we can lower that interaction. We can't eliminate it entirely. On top of that, this final negatively charged layer, it's usually, we choose a dense negative charge and a molecule that is very brushy, gives us a nice kind of shag carpet root structure on the surface, you know? And that gives us steric repulsion, meaning that if something lays on its surface, it's sterically going to be moving about and wants that freedom, and it will shake that protein off. So we get a little bit less of that undesired protein absorption. So we call this the stealth layer. You can also think of this as a layer that has a lot of water molecules associated with it, a lot of negative charge, and it looks more like something that belongs in the bloodstream. And so we're able to survive for a longer time period. This is what these look like. On the left, you can see a liposome, which is, you know, lipid bilayer surrounding water, essentially. A sphere that surrounds water. And inside, we can have a water-soluble drug. We can layer it, as you see on the right, with our layers, and it doesn't change it very much. It's a little bit more solid, but it's not too much bigger. Maybe about 20 to 30 nanometers bigger. And on average, these are about 100 nanometers in size. You can also layer polymers, so PLGA or polystyrene nanoparticles. And I don't know if you can see it here, but there's kind of like a halo. You can see the layered film around the solid nanoparticle on the right. You can layer a bunch of other things, lipid nanoparticles, vesicles, a range of things can be layered in this way. Now I'm trying to think if you can see color as well if I need to turn down the lights. I don't want to put people to sleep. But you could turn down the lights if I figure out how to turn this on. I will leave it alone, but let me know if you're having trouble seeing. What we found is that when we deliver the drugs, we can actually take advantage of that outer layer in more than one way. Along with being a stealth layer, we found that some of the negatively charged polymers that we use also have a very natural affinity for the cancer cell. So hyaluronic acid, how many of you have heard of that before? Multiple, right? And maybe in a L'Oreal product or something like that, right? It's a very, very hydrating polymer, strong negative charge. It's a natural polysaccharide, and it's a part of the natural part of the matrix of our tissues. But it turns out that it's also a ligand for a receptor called CD44, and that receptor is highly overexpressed and a large number of tumors, because these tumors are growing rapidly. Some of their genetic mutations allow them to produce these CD44 receptors so they can grab onto hyaluronic acid and essentially grow more rapidly. So we found that cancer cells take up our nanoparticle much more than non-cancerous cells. You can kind of see the red glow here. We also found that our nanoparticles can penetrate through the stromal layers, which are the blue layers of the tumor, and accumulate in the core of the tumor. And here, the nanoparticle is green, and CD44 labeling is red, and this orange-yellow is the area where the nanoparticle is accumulating in tumor cells. So in our earlier work, we were looking at models that we understood fairly easily, and then we moved to more difficult representations of cancer in animals. In this case, we're working with our friend Tyler Jacks, who's another faculty member at the Koch Institute in Biology. And he had a mouse model that had sort of naturally occurring, really aggressive version of non-small cell lung cancer, which is a really aggressive form of lung cancer. And it has some unique genetic mutations. What we found is that when we injected our nanoparticle into healthy mice, we would see the whole mouse light up because the nanoparticle has a half-life in the blood of about a day. So the mouse is sort of lit up, all of the blood contains those nanoparticles. And then after that, you can see where they remain, and they get filtrated through the liver, but otherwise they don't light up other organs. When we look at the mice that have the lung cancer, we find that there's a lot of accumulation, not just in the liver, where it gets excreted, ultimately, but in the lungs. In fact, there's a 40 to 50-fold higher accumulation in the lungs, which suggests that we are targeting the tumors there. And if we stain for CD44, that brown stain you see here, we see indeed that there is a lot of CD44 in the body. I mentioned that this kind of cancer has some really specific genetic mutations that make the cancer highly aggressive. One of them is an uncle gene that's called KRAS, and we can silence it with siRNA. Another one is a missing gene called P53. It undergoes a mutation. And when that mutation is gone, it basically allows cancer to grow more rapidly, because P53 is a monitoring gene that causes cells to undergo a kind of kamikaze cell death when there's too much DNA damage. Without P53 present, if you use a DNA damaging drug, cancer cells won't direct themselves toward death. So we decided to replace that gene with a microRNA known as miR34A. So our layer by layer blue film, the nucleic acid film, contained KRAS siRNA and miR34A. So when we actually tested the model, here we're using a model where the tumor doesn't glow. So we had to use micro-CT to see them, and maybe you can see little red circles where my student was pointing out where the tumor exists. We found that tumors were much smaller when we had the combination of both the drug, which was cisplatin in the core, and the siRNA. And we also found, when we looked at how long the animals survived, this is a survivability curve, we compare how rapidly they died within the first 10 days compared to how long we can keep them alive with the combination, which is going out about 3 times longer and gives us about 30% more survivability than if we just have the chemotherapy drug and the nanoparticle. So this kind of approach is really exciting to us, and we continue to look at it for other kinds of cancer, including lymphoma and glioblastoma. However, one of the areas we really focus on in our lab is ovarian cancer. And one of the reasons for that is that among the different cancers that affect women, ovarian cancer has had perhaps the least amount of progress. We've had some great work done in breast cancer, in which we found some critical genetic changes that allow us to target it. But in ovarian cancer, it's been harder, and there are reasons for that. First of all, ovarian cancer has very subtle changes that occur. So it's detected really late. Typically, it takes a while for the doctor to get it right. And by that time, we're at the third or fourth stage of the cancer. On top of that, ovarian cancer tends to respond to treatment the first time. We get like 70% response, and that treatment is something like a really hardcore chemotherapy drug, like the cisplatin I just named, or paclitaxel. But after that hardcore treatment and surgery to remove the tumors, about 85% of patients have a recurrence within the next 5 years. And that recurrence is usually a really resistant form of ovarian cancer that doesn't respond to those drugs anymore. So this is really a difficult cancer to treat. It doesn't have a signature genetic mutation. We can't just say, let's do KRAS or let's replace P53. It has multiple different mutations depending on the patient. And it also tends to be sneaky in that it's not the same in every patient. So this is a great opportunity maybe for an immunotherapy where you can train the body to attack the cancer when it comes back. Unfortunately, the most common immunotherapies have not been effective in ovarian cancer. And the rationale around that is that there aren't a lot of immune cells that are present in the cancer or the tumor that are able to be activated or impacted by the known immunotherapies. Okay, so when we started thinking about how do we treat ovarian cancer using this layer-by-layer approach, my former student Santi, who's now a professor at Columbia, at that time was working with 2 undergrads, Arya and Ben, and looking at how we might be able to find a nanoparticle that has a lot of adhesion and association with ovarian cancer cells. And we did this first by just looking at a library. We took a negatively charged liposome. We observed poly-L-arginate, which is a positively charged polypeptide. And then we absorbed a number of different negatively charged polyamides. We went to Sigma, we ordered a bunch of known negatively charged polypeptides, and we just tried them. And out of that group, there were 3 of those polyelectrolytes that were particularly interesting. 2 that we've been working with a great deal to address ovarian cancer. And one you already know from our talk before is hyaluronic acid. Hyaluronic acid, when the particle is taken up, it's taken up inside of these little vesicles. You can see the green. They're moving in and out of the confocal microscope. They get into these endosomes, and eventually they break out through endosomal escape and release the drug. Great for delivering siRNA that needs to get into the cytoplasm. Great for chemo drugs, where you need to get to the nucleus ultimately. Poly-L-glutamic acid is different. It has a high affinity, but as you go through the screen, you can see that it sticks to the edges. It sticks to the outsides of the cell. So we have these green nanoparticles. They're sitting on the edges of the cancer cell, and they will stay there for hours, and sometimes even a few days. And then there's this in-between one, poly-L-aspartic acid. It internalizes really slowly and through a mechanism that's different from hyaluronic acid. So we recognize it goes into slightly different compartments. We took a close-up on hyaluronic acid and poly-L-glutamic acid. For both of these, we found that if we were to look at a mouse model where we have ovarian cancer that is present in the abdominal cavity, so typically ovarian cancer starts in and around the ovaries but spreads across the abdominal cavity, we can label the tumor, luciferase, and the nanoparticle here has an IR signal, and this is nanoparticle that is injected directly into the abdominal cavity. We're literally sort of painting the tumor with the nanoparticle. These are sticking to the tumor with great faithfulness, and you can see the overlay. Even when we have metastases, like this met to the intestine, we see that the nanoparticle goes where the metastases are as well. So you really have a system that is faithful to where the tumor is, regardless of where it's gone. So we thought, okay, we can start with this. With hyaluronic acid, we have collaborations, one of which is with Joan Brugge, who's at Harvard Medical, and Ursula Machulonis, who is the head of gynecologic oncology. I spent a lot of time saying that word and getting it out correctly at Dana-Farber. And in this work, she had these two small molecule drugs. I know some of you are thinking about pharmaceutical chemistry, so this is all cool looking at these structures. But these are two inhibitors. They each are a little bit toxic, but not too bad, and they can be delivered orally. But together they're super toxic, they synergize, and so you can't give a patient these two together. And Joan's question was, can you incorporate them in a nanoparticle and kill the cancer without hurting the patient? So we did work in which we put them in two different nanoparticles, we put them together in one nanoparticle. We found that the one that has both drugs together is much more powerful. And Stephanie Kahn, who did this work, found that when she administered those combination nanoparticles, the tumor, here glowing sort of masses in the mouse, are much smaller in the combination nanoparticle over time, and there are very few tumor lesions that grow in the mouse. So that's chemotherapy, but as you know, chemotherapy is good for a first round, but not when we get recurrence. We have a collaboration with Daryl Irvin, who is an immunobioengineer. And we talked about this nanoparticle that presents things on the outside. This would be great for presenting a protein to a neighboring cell, as opposed to delivering the protein to the cell, right? Kind of hold it out to your neighbors. And we talked to him about what might be useful. And he mentioned the cytokine, IL-12. The cytokines actually activate the immune response and lead to an inflammatory response that can help to address cancer. So the idea was we can take this very powerful cytokine known as IL-12. It has a special interaction with T and NK cells, the two cells that are really responsible for killing tumors, as well as the inflammatory response we need at the beginning, which is going to cause immune cells to generate interferon gamma. How many of you are familiar with interferon? Any yet? I see a few. Yep. That interferons are what get the immune system going. And if we get the immune system going near the thing that you want to have the immune response against, you can actually get a very directed response. So if we get this going in the tumor has all of these antigens that are just naturally present. And now we have an immune inflammation, kind of an inflammation event. We're going to train those cells to attack that tumor. So IL-12 is interesting for that reason, but it's really toxic. Because as you can imagine, if you just deliver it to your body, you will activate immune responses against your own tissue. And this is what happened when they tried to use it in clinical trials. There were these huge sort of full body responses, which were dangerous. And in fact, there was loss of one life. And that killed the use of free IL-12 for delivery. So the idea here was, can we introduce IL-12 in a nanoparticle? And the nanoparticle will protect from interactions with other cells. If we can get that nanoparticle to go to ovarian cancer cells, then we can activate the immune response against it. So here's how we developed this system. We take a liposome, you can see it a little better here, bilayer, and some of those lipids have negative charge. We use a lipid that has a conjugatable group at the end, and we attach IL-12 protein to the lipid, and now we have some IL-12 conjugated directly by lipid to the liposome. Because it's negatively charged, we can absorb the poly-L-arginine and then poly-L-glutamate. So So we have the unlayered liposome, and we see UL, we're referring to this guy. And then we have the IL-12 conjugated system, which has PLV on the outside, poly-L-glutamic acid. And the whole idea was, let's have this hang out on the surface of the ovarian cancer cell. There aren't a lot of immune cells present, but the ones that are present will engage with the IL-12 in some way. And we can then activate the immune cell, and it will signal with interferon gamma more immune cells, and we'll start a cascade that will lead to a change in that immune microenvironment. Here's some chemistry. All right. So the conjugation that we first used was a nickel histidine. That basically uses the non-covalent interaction of nickel with histidine to get this protein anchored on. And it's used a lot in biochemistry as an easy tag. However, we can't take it in the clinic, but also it has some side effects. But also, it releases over a time period of about 24 hours. But we thought, let's start with this, and we'll release the IL-12 to the immune cell. The other system we used was a conjugation using a thiol-malleamide linkage. All right? So am I talking the language of some of you here? So this is a nice covalent bond. It's very, basically we can get a very high conversion. So we have two bonds, one is temporary and one is permanent. And this is work done with Ivan and his undergrad Ezra. So when we look at how much we're able to, engage these IL-12 in the tumor, we can measure the IL-12 by labeling it. And here we're looking at IL-12 in just the free form, how much of it is present in the tumor, in tumor mice. And we're down here. Now, if we look at the nickel in the unlayered system, we're releasing it. After 24 hours, not much left. If we layer it, then it's sticking but still releasing. So after 24 hours, a little more. If we use the malleamide, which is the permanent linker, but we don't have the layers, it's not sticking. And so we have some happening here, but it's still not impressive. But if we have both the malleamide permanent layer and we have the layered film, we see that we have something like 8 to 10 times more of the nanoparticle with the IL-12 present at the same time point. So we are associating with tumors, and we're hanging around. Even after a week, we can see that the malleamide LBL system has about 4 times more presence than the other formulations. So IL-12 sticks around. Sticking around is important in an immune response because you want constant stimulation to cause those immune cells to arrive and begin to inform the T cells of what to attack. The longer we do this, the more we're able to engage T cells that will remember what it should attack. Speaking of T cells, we can measure how many T cells are present in tumors. And we use this using cell sorting or FACS. On the left, you see kind of the output of FACS. How many of you have worked with that tool or seen someone work with that tool before? I see a few hands. Exactly. It actually is a method of we take the tumor, we dissociate it, and then we put those cells through a machine that measures whether or not there's a signal. And the signal we're looking for is associated with the identity of the cells. And we can count how many cells have markers for T cells. In this case, with just free IL-12, the markers for T cells would appear in this square window, and there's none there. The same with the unlayered nanoparticle and just the control, which is just sugar, dextrose. But with the LBL nanoparticle, we see that there is a very meaningful population of cells that are associated with T cells. If we count the number of T cells, this is a quantitative experiment, we can see that there are something like 7,000 to 8,000 T cells compared to the other systems. Finally, there are different kinds of T cells, the kind that kill and the kind that kind of support. CD8 positive T cells are the ones that kill, and we'd like to have more of them present. So if we look at the fraction of CD8 positive versus CD4 positive, we can see that this is a negative log fraction until you get to the LBL nanoparticle, and then you see that there's actually a greater fraction of CD8 positive over CD4 positive. More of the killers than the helpers. If you then treat an animal that has a very advanced metastatic form of ovarian cancer, it's a very aggressive model, with just one injection of our IL-12 nanoparticle, what happens? So here we're looking at a 55 microgram dose 7 days after the occurrence or introduction of the cancer. And we're looking at how long the mice survive, where the dotted line is the untreated mouse. And the free IL-12 is this green line here, where you get some mice surviving, but the rest die pretty rapidly. And we found that, in particular for this formulation, the mammalian LBL IL-12, we are able to see an extension of survivability. For those that did survive, we can challenge the mouse again to see if the immune system is going to attack the tumor. And we found that when we introduced new tumor, new cancer cells, all of those mice that survived the first time survived the onslaught of cancer the second time. And this is that memory that we're looking for. But I mentioned that one of the real difficulties with ovarian cancer is that you don't have a lot of immune cells to begin with. And that's why they don't respond to really common immunotherapies like checkpoint inhibitors. How many of you have heard of that term, checkpoint inhibitor? I see a couple. Yeah, it's basically a small molecule drug that suppresses a kind of regulatory response that cells have to turn off the immune response. So we want to use the checkpoint inhibitor to stop that from happening. They're not really effective in ovarian cancer because there's nothing happening in ovarian cancer. So we thought we've changed the environment, maybe a checkpoint inhibitor will be relevant now. So we combined the IL-12 with the checkpoint inhibitor. And what we found was that the mammalian LBL nanoparticle system gave us 100% survivability of the mice after just 2 injections of the combination. And this we're really excited about because, well, for one thing, 200 days is a long time for a mouse. We found that we tested those mice, and they all survived when we reintroduced the cancer. So these are mice that were able to survive recurrence of cancer. And now we're looking at how we might be able to translate this. So a couple of questions you might ask about what's going on here. I said polyglutamic acid. Why does it do what it does? I mean, it's sticking to the outside of the cancer cell membrane. We did not know for the longest time. So Ivan had to take apart this question by asking several smaller questions. What does polyglutamic acid bind to? And we couldn't at first think of any kind of cancer cell receptor that had a specific interaction, but over time we think, well, look, this is a polyamino acid, right? It's just a repeat of glutamic acid again and again. So maybe it's an amino acid type of thing that's going on. This is glutamate. And maybe there's some sort of amino acid transporter. These are the proteins that sit on the cancer cell surface or any cell surface, and they move amino acids into the cell. It's an essential part of any cell's metabolism. So in this work, I then asked the question, can glutamate be responsible for this because it's binding to the glutamate amino acid transporter? He rapidly found that glutamate amino acid transporters aren't really that highly expressed in cancer cells. However, glutamine, you know, like the cousin of glutamate, is something that is highly overexpressed in a lot of cancer cells. There's something called glutamine addiction. The way the cancer cells grow, they metabolize a lot, they're consuming a lot of energy, and glutamine is one of the key components in metabolism. And this, hats off to my biological friends, is like a metabolic diagram of what happens, you know, but the key issue here is that because the cancer cell needs a lot of glutamine, it puts out a lot of these amino acid transporters. And it turns out that if we look at glutamate and the binding constant for glutamate versus glutamine, when glutamate is in its slightly acidic form, it actually can outcompete glutamine. So we think that in this brushy form of polyalglutamic acid absorbed on the surface, we have something that is binding the glutamine transporter. And Ivan did do work to show that indeed that is what happens. He can systematically block the glutamine receptor transporter and find that he gets less uptake. And he can use siRNA to knock it down and find that nanoparticles aren't taken up as much. This is part of a much bigger project where we were trying to understand why this system works so well. There are other aspects of it, and this is a cartoon that shows everything from the accumulation in the tumor to the transport within the tumor, and the way in which these IL-12 systems that are covalently bound, they ultimately actually exchange with lipids on the surface of the cancer cell. And we found that because they have a lifetime of 4 to 5 days, it's long enough to begin to decorate the cancer cell with the IL-12 protein. So it's something that we didn't anticipate, but this particular approach allows us to decorate cancer cells with the IL-12 itself, the therapy that we actually need to stimulate the immune response. Okay, so what I might do, because I don't want us to go overly long, is give you one short example of how we used this approach for brain cancer. I will say as an aside that we have been working on incorporating mRNA, and we've been layering lipid nanoparticles that contain mRNA to target ovarian cancer in much the same way, but we're training the cells to generate the protein on their own. In glioblastoma, we had a similar sort of challenge. We wanted to understand how we might be able to design a nanoparticle that could address some of the challenges of glioblastoma. And one of those biggest challenges is getting past the blood-brain barrier that protects the cells in the brain from toxins. And we also needed to be able to essentially diffuse through the tumor. So we actually started working on nanoparticles that could be delivered using an approach called convection-enhanced delivery, where you essentially just think of pushing the liquid into the tumor. It pushes it past the blood-brain barrier by convective force.